Assalamu alaikum. Dear students, I am here Hafiz Muhammad Ammar, co-teacher for course Microbiology, code BOTN 4128. Today's lecture number 18, we will study some other pure culture techniques except streak plate method. Streak plate method we have discussed in previous lecture. So in today's lecture, we will focus on pour plate method, serial dilutions and spread plate methods. The basic principle in the pour plate method, that is to dilute the inoculum in successive tubes containing liquefied agar media to a thorough distribution of bacterial cells within the media. Pour plate method. Another way to obtain pure cultures, the pour plate method, makes use of serial dilutions. A series of dilutions are made such that the final dilution contains about 1000 organisms. Then 1 ml of liquid medium from the final dilution is placed in 9 ml of the melted agar medium, which is about 45 degrees Celsius temperature, and the medium is quickly poured into a sterile plate. The resulting pour plate will contain a small number of bacteria, some of which will form isolated colonies on the agar. Because this method embeds some organisms in the medium, it is particularly used for growing microaerophiles, those which cannot tolerate exposure to oxygen in the air at the surface of the medium. If the material to be tested is an aqueous liquid, a known volume is simply placed in the face of the petri dish at 10 to 25 ml of molten culture medium, typically a tryptone soya agar or plate count agar at 45 degrees Celsius temperature, is poured onto it and quickly mixed by gentle swirling. Then the plate is placed on one side for the agar to set. If the sample is a solid that is soluble in water, that solid would normally be dissolved. But if it were insoluble, then a suspension would be used. The problem in the last case would be to ensure that the suspension remained uniformly dispersed during the pipetting and any dilution steps. Because the sample is dispersed through the medium before the gel sets, the colonies that grow are similarly dispersed throughout the agar. Consequently, the colonies are usually of different sizes because there is less oxygen available within the gel than its surface, and most organisms are either strict aerobes or facultative anaerobes, which will grow best where the oxygen concentration is highest. This technique is used to perform viable plate counts, in which the total number of colony forming units within the agar and on the surface of the agar on a single plate is enumerated. Viable plates counts provide scientists a standardized means to generate growth curves, to calculate the concentration of the cells in the tube from which the sample was plated, and to investigate the effect of various environments or growth conditions on bacterial cell survival or growth rate. There are the following advantages of this technique. It is easy to undertake and will detect lower concentrations than surface spread methods because of the large sample volume. It requires no pre-drying of the agar surface as it is required in other culture methods. The most common method for determining the total viable count is the pour plate method. The pour plate technique can be used to determine the number of microbes per ml in a specimen. It has the advantage of not requiring previously prepared plates and is often used to assay bacterial contamination of foodstuffs. The use of relatively hot agar carries the risk of killing some sensitive contaminants, so giving a low result. Small colonies may be overlooked in the case of solid sample dissolving in water. Some species may suffer a degree of viability loss if diluted quickly in the cold water. Consequently, isotonic buffer, phosphate buffer saline for example, or peptone water are used as solvents or diluents. Colonies of different species within the agar appear similar, so it is difficult to detect contaminants. The reduced growth rate of obligate aerobes in the depth of the agar. The preparation for the pour plate method is time consuming compared with the streak plate and or spread plate technique. Serial dilution. As the name suggests, a series of sequential dilution that are performed to convert a dense solution into a more usable concentration. In simple words, serial dilution is the process of stepwise dilution of a solution with an associated dilution factor. In biology, serial dilution is often associated with reducing the concentration of cells in a culture to simplify the operation. The basic principle of serial dilution is to inoculate a series of tubes with a microbial suspension so dilute that there are some tubes showing growth of only one individual microbe. This method is commonly used to obtain pure cultures of those microorganisms that have not been successfully cultivated on solid media and are being grown only in liquid media. A microorganism that predominates in a mixed culture can be isolated in pure form by a series of dilutions. The inoculum is subjected to serial dilution in a sterile liquid media, and a large number of tubes of sterile liquid media are inoculated with the aliquots of such each successive dilution. The objective of the serial dilution method is to estimate the concentration of an unknown sample by enumeration of the number of colonies cultured from serial dilution of the sample. Air concentration may be the means of number of organisms, bacteria, viruses or colonies. So in serial dilution, the density of cells is reduced in each step so that it is easier to calculate the concentration of the cells in the original dilution by calculating the total dilution over the entire series. Serial dilutions are commonly performed to avoid having to pipette very small volumes, even 1 to 10 microliters, to make a dilution of solution. By diluting a sample in a controlled way, it is possible to obtain incubated culture plates with an easily countable number of colonies, around 30 to 100, and calculate the number of microbes present in the sample. The sample culture is taken in a test tube and 6 test tubes each with 9 ml of sterile diluents, which can either be distilled water or 0.9% saline, are taken. A sterile pipette is taken. 1 ml of properly mixed sample culture is drawn into the pipette. The sample is then added to the first tube to make the total volume of 10 ml. This provides an initial dilution of 100. The dilution is thoroughly mixed by emptying and filling the pipette several times. The pipette tip is discarded and a new pipette tip is attached to the pipette. Now 1 ml of mixture is taken from the 10 minus 1 dilution and is emptied into the second tube. The second tube now has a total dilution factor of 10 minus 2. 10 power is of minus 2. That you can say ratio of 1 into 100. The same process is then repeated for the remaining tubes, taking 1 ml from the previous tube and adding it to the next 9 ml diluents. As 6 tubes are used, the final dilution for the bacteria cells will be 10 power is 6, means 1 into 1 million. Another explanation. For convenience, suppose we have a culture containing 10 ml of liquid media containing 1000 microorganisms. 100 microorganisms per ml of the liquid media. If we take 1 ml of this media and mix it with 9 ml of fresh sterile liquid media, we would then have 100 microorganisms in 10 ml or 10 microorganisms per ml. If we add 1 ml of this suspension to another 9 ml of fresh sterile liquid media, each ml would now contain a single microorganism. Even though serial dilution is a useful technique in labs, it faces some challenges, some of which are: an error might occur during the propagation of the sample, and the transfer inaccuracies lead to the less accurate and less precise transfer. This results in the highest dilution to have the most inaccuracies and the least accuracies. Because serial dilution is performed in a stepwise manner, it requires a more extended period of time, which limits the efficacy of the method. Serial dilution only allows the reduction of bacteria, but not the separation of the bacteria, like in other techniques like flow cytometry. This technique also requires highly trained microbiologists and experts in aseptic techniques.
In this practical, we're going to give you a contaminated water sample, and one of the things we want you to find out is just how many bacteria it contains. You might think this would be as easy as just plating out the contents onto an agar plate and seeing how many colonies grow. However, if we do this, what will probably happen is there'll be too many colonies to count, and if the colonies grow into each other, then we can't count them accurately. We therefore need a way to dilute the sample enough so that we can count individual colonies and work out how many colony-forming units were present in the original sample. Remember, not all bacterial cells in the sample will be able to grow on this particular type of agar, so you should call them colony-forming units or CFUs throughout. Don't call them cells because the two things aren't quite the same. We're going to do this by setting up a serial dilution. The basic principle is we're going to set up a series of tubes, each of which contains a more dilute version of the sample. We'll then plate each different dilution onto a different agar plate and allow the bacteria to grow overnight. Then hopefully one of those plates will have between 20 and 100 colonies, which is a good number to count. We can then work out from this system how many colony-forming units were present in the original sample. To do this, we're going to make up several tenfold dilutions. So this means we'll add one part of the sample to 9 parts of a diluting liquid so that the new tube is 10 times more dilute than the previous one. So for example, if the original sample had 100,000 colony-forming units in it, the new one should have 10,000 colony-forming units. We'll call this the 10 to the minus 1 dilution. 10 to the minus 1 means exactly the same thing as 1 divided by 10, but biologists usually express dilutions in the exponential notation, so make sure you're happy with this. In our case, we'll take 0.1 ml of the sample and add it to 0.9 ml of sterile nutrient broth, and this is our first dilution. However, if we play this dilution out, we might still have too many colonies to count, so we're going to have to set up another dilution. The easiest way to do this is to do exactly the same thing again, but this time to dilute the 10 to the minus 1 dilution by tenfold to give a 10 to the minus 2 dilution. So we'll take 0.1 ml of the 10 to the minus 1 dilution and add it to a fresh 0.9 ml of sterile nutrient broth. So this has now been diluted 100 times compared to the original sample. So we'd expect this sample to have 1,000 colony forming units in it. We can keep doing this again and again to set up a series of dilutions. In the practical, you'll make dilutions from 10 to the minus 1 to 10 to the minus 4. You'll then spread 0.1 mils of each dilution onto a separate agar plate. In the second practical, you'll then count the colonies that grow on each plate. Remember, you're going to need to have between 20 and 100 colonies on at least one of the plates to get an accurate indication of the number of colony forming units. Any more than that, and there'll just be too many colonies to count, but counts below 20 tend to be quite inaccurate. It just becomes a matter of chance as to whether you get, say, 4 or 6 bacteria on the plate, but that might mean that you get quite significant errors in your calculations. Make sure you've understood how
The separate plate method is a technique to plate a liquid sample containing bacteria so that the bacteria are easy to count and isolate. A successful spread plate will have a countable number of isolated bacteria colonies evenly distributed on the plate. Spread plate culture technique is among the most widely used culture technique for isolating the bacteria. Basic principle: when the media plate is spun at some stage, single cells will be deposited within the bent glass rod spreader onto the surface of the agar media. Some of the cells present in the specimen, diluted specimen, will be separated from each other by a distance sufficient to allow the colonies that develop to be free from each other. Method of separate plate technique: first, make a dilution series from a sample. Pipette out 0.1 ml from the appropriate desired dilution series onto the center of the surface of an agar plate. Dip the L-shaped glass spreader into an alcohol. Flame the glass spreader over a Bunsen burner. Then spread the sample evenly over the surface of agar using the sterile glass spreader, carefully rotating the petri dish underneath at the same time. Incubate the plate at 37 degrees Celsius for 24 hours. Then calculate the colony forming units value of the sample. Once you count the colony, multiply by the appropriate dilution factor to determine the number of colony forming units per ml in the original sample. The separate plate technique, in conjunction with the serial dilution, is a valuable research tool. To demonstrate the cultural characteristics of the bacteria like color, texture, size, elevation, etc. To isolate the bacteria in discrete colonies from the specimen containing more than one bacterium. For determining the sensitivity and or resistance of the bacteria towards a particular drug or antibiotics or a test substance, to obtain the sufficient growth of the bacterium from various biochemical and other tests. This technique is also used to estimate the viable counts of the bacteria in the specimen, and it also used to maintain the stock cultures, to transport or short-term storage of the specimen like stab cultures, etc. The main advantage of the separate plate method is the heat sensitive microbes are not affected in this technique, and no subsurface colonies appear in this method, so isolation of the organism is more easier. While if we talk about disadvantages of this technique, strict aerobes are favored while microaerophilic tends to grow slower. Crowding of the colonies makes the enumeration difficult. Accurate dilution using pipette should be made. Volume no longer than 0.1 ml can be spread on the nutrient agar plate. Here is the difference between pour plate method and separate plate method. You can see in step 1 the inoculation to be made in empty plate. while inoculation in the spread plate method is containing a solid media. In the pour plate method, melted nutrient agar is added and swirl to mix up the culture, while in the spread plate method the spread inoculum is being spread over the surface evenly. The fourth one is colonies grow in and on solidified media in the pour plate method, while in the spread plate method colonies grow only on surface of the media. Here are the references where from this lecture was prepared. You can also study these reference books or articles. Thank you so much.